I am the Chief Medical Officer of Natera, the panorama test that I'm about to talk about, and I hope one of the goals is for you to see why many of us really are so passionate about using SNPs to move the field forward. This is a busy slide, and it's supposed to be busy. This actually explains what Dennis did for the field. On the side of the screen, this is a cut and paste from ACOG. In 1983, we found the first protein marker in maternal system that set you at risk for aneuploidy. And the reason it's busy is because every few years, and this was part of the research I did, we tried to find more markers, we tried to use ultrasound, we tried every which way to make this better. And in fact, this is the end result. After a tremendous amount of effort, 5% false positive rate. So I want to just set the clinical baseline here. In a practice, and in California, it might be up to 7% of women will get a phone call. You had a blood test, Miss So-and-so. You're no longer normal risk. You're now high risk. You have to come see someone like me who does fetal medicine. And out of all the women that we sent for amnio, only 1 in 20 actually had the problem. You just heard Dennis's talk, completely changed the world, really almost overnight. The counting method that Dennis mentioned to you was quite brilliant in what you see here. You just look at sequences of DNA that are of interest, for example, chromosome 21 pieces, and you just count. If you have more, or if you have the usual amount. So if it's the amount that you expect to see, let's say of 13, or pieces of 21, you're screen negative. If you have additional pieces, you're now screen positive. And that's the genius. It had to be better than what we had. In the old days, we looked at beta HCG and other protein markers that in fact were a proxy for the placenta. The innovation here was actually looking at what you care about, which is DNA from the placenta, and that you could actually measure and count it. Let's talk about SNPs. And what really is the next wave here, single nucleotide polymorphisms. Many in the audience, of course, know this very well. And it's when you look at the actual base pairs, not the entire sequence. So if somebody is sitting there and they have a base pair of T, the person beside you is a C, you are no longer the same person. Okay, it's a remarkable thing. It thankfully happens every few hundred times. In the sequence, it is sequence diversity in a single species, that is human beings. Again, we have some of the top scientists in the world in the room today. They all know that they use this every single day, all over the world, to differentiate individual genomes and genotypes. But even people who never set foot in a lab know this. We don't use fingerprints anymore when we do forensics. If there's a crime scene, what do you use? You use genetic fingerprinting. It is incredibly accurate. And the way the test is done is you take a blood sample from the mom. So this is using the panorama test as opposed to the other approaches. Spin the blood, get the mother's sequence, and then go back to the plasma, get the mix, and then a very complicated algorithm actually pulls out the fetal DNA signal. And that's how the mom gets her personalized risk score. And this is the case I want to make today. We are at the Personalized Medicine World Conference. We heard this morning how you think about personalized medicine. And here's just one definition, but it's essentially the same. Prevention, detection, and treatment of disease, taking into account a person's unique genetic profile. In prenatal medicine, what I'm asking you to think about today is we must assess the unique genetic profile of the fetus in question. That's when we'll have hit the bar that we really are talking personalized medicine. The constellation metaphor. Every night you look up at those stars. It was actually a beautiful night last night in Palo Alto. The ability to pull out what you want to see. Astrophysicists have been doing this forever. It's signal to noise analysis, and our eye does it also when we see constellations. So SNPs have that ability to pull out unique genotypes. And it is a unique approach. And in my few minutes left, what the case I want to make here is it's innovative at 2 points. The clinical landscape, and especially important, the ability from a business point of view to develop a global footprint. So again, a few minutes left. I'm going to go through this relatively quickly, but the NIPT tests, are they all the same? Do they all do the same thing? And I think you just heard from Dennis. You actually have now to think about distinguishing different haplotypes and different genotypes. The issue of SNPs, it corrects two fundamental problems with counting methodology alone. Okay, what are the problems? It's one problem, actually, it's called discordancy. What happens when the serum screen says one thing, but the outcome is different? That's discordancy. It happens in two different ways. False negative, Miss So-and-so, no worries, your screen test is negative, and then baby's affected. Or the other way, Miss So-and-so, like I said, the phone call, screen test positive, you gotta go see somebody like Dr. Gross, you may have an invasive test, it has inherent risk, et cetera, et cetera. So today, what I'm going to do actually is just focus on the false positives, even though SNPs help correct the false negative problem as well. So what happens in the case of a false positive? You have the situation of screen positive, but outcome normal. There's 3 causes reported in the literature, basic causes, if you use counting alone. When you look at total amount of DNA and just count the sequences, what are the 3 things? First of all, mother and not the fetus has an abnormal chromosome. Next is additional fetus, which I'll explain at more length, that's far more serious. But the placenta does not adequately reflect the fetus. You just saw very elegant research by Dennis indicating clearly that what is floating around in the plasma is actually not fetal, it is placental. When we all started our early lives, half our cells became us, half our cells became our placenta. And therefore, very, very similar. It is not perfect. And I want to state very clearly, this is why non-invasive in its current configuration cannot be a diagnostic. There will always be that slight difference, which is why we always tell women, even with an excellent non-invasive test, to consider confirmatory testing. So let's start with the mother is the issue, and not the fetus. So this is actually very real. This is a wonderful paper using counting methodology. 8.6% of the time, if there is an abnormal X chromosome on the screening test, the mother had the abnormal X chromosome. Just think about that. It's actually fantastic. These are happy, healthy women. Okay, this is normal biology. I'm a medical geneticist. This is always on our board exams. It's a very famous graph. As women age, their X chromosomes drop out. I can promise you that I actually am a mosaic Turner syndrome. It's absolutely normal. And in fact, in my genetic lab, I would read out that as low level being normal. Okay, so this is what's happening. If you use total and you can't individualize, you're actually picking up an issue with the mom. Now, the second issue, however, is much more serious, and this is where you have to use SNPs, at least for now, that telling the difference between mom and fetus is good, but what if there's another fetus or another set of chromosomes floating around in the uterus? How can you tell them apart? The additional fetus problem is known as the vanishing twin conundrum. This is how it works, and we know this from all the excellent IVF data. Many of us actually started out as a twin pregnancy, and then one twin vanishes early on. Usually that twin has an issue. So we can set up very easily. Normal female, and then the twin is a trisomy 18, terrible disorder, vanishes, early miscarriage. If you only can pull out of plasma total DNA, what is the call going to be? It may be trisomy 18, except the fetus is actually normal. Or try this. What if the vanishing twin is a male, but the fetus that remains is a female? Those Y sequences will get detected in plasma. It's going to be called a Y. That's where gender discordancy comes from. This is our paper that was recently published, and the conclusion says it all. The ability of SNPs to identify additional fetal haplotypes is expected to result in fewer false positive calls and prevent incorrect fetal sex calls, and is in fact now been demonstrated over this past year several times in the literature. And so just to finish, I have just 1 minute left, but let's go back to our constellation, because I promised you to show you why this makes sense from a business development point of view as well. You can actually use the cloud to pull out the DNA and use a cloud-based DNA platform, which we're actually calling the Constellation DNA platform. And again, if you use this SNP approach, again, many of the expert scientists will tell you it is not a tour de force to use sequencing to look for 20,000 SNPs, which is basically what the algorithm currently works with. It truly isn't. The tour de force actually happens when you look at the cloud. It's the algorithm, okay? And we just heard about it, right? Software, DNA, and the connection between DNA and software, which was a wonderful concept. So in other words, any lab should really be able to run a SNP assay, 20,000 SNPs, using sequencing. You can upload. It's not a lot of, you can upload the data, and you use the Natera cloud-based software, and then you can send the copy number and QC results back to the lab. This is how you can readily move out and really develop a global footprint. And just to finish my last slide here, the concept of the constellation, right? The way you look at the night sky, and again, astrophysicists have been doing this well, but that you can pull out the signatures, you can pull out the signals. The constellation software is also delivered through the cloud. It's scalable. It's readily scalable. And again, just to finish off now, the SNPs, it's not just non-invasive prenatal testing. At Natera, that's just one product. But any product where you have to be able to tell needle in a haystack and do this individualization of genotypes, you can imagine would work very, very well, whether it's miscarriage testing, pre-implantation genetic diagnosis, forensics we spoke about, research, transplantation, and now most important, as Natera works towards liquid biopsy in cancer. Because in fact, the placenta, as we know, it's a tumor, right? It's a growth in a sense, and where the genotype actually doesn't match the mom.